We are still talking about the cell cycle. We've gone through the details of interphase. Now we need to talk about what happens in M, the phase called mitosis. Mitosis is going to be getting the cell ready to divide and then pulling those sister chromatids apart. Remember, in G1, all of the chromosomes consisted of just one chromatid, just one piece of DNA for all 46 chromosomes. In the S phase, we replicated that DNA, and now we have a sister chromatid attached. They're genetically identical to each other. We have 46 of these as we move into G2. Now the goal is to pull these two identical pieces of DNA into two separate cells so we can make two new genetically identical cells. And that's what we're going to do in the M phase. This M phase really includes two parts. Division of the nucleus, which is really officially what mitosis is referring to, and then cytokinesis, which is going to be division of the cytoplasm to make two new cells. And then each of these cells immediately goes into G1, and the clock just keeps going. So let's talk a little bit more about the details of what happens during the M phase of mitosis. So mitosis is typically divided into these phases: prophase, prometaphase, metaphase, anaphase, and telophase, also called telophase. Cytokinesis happens sometime in here. It gets complete at the end of telophase. That is division of the cytoplasm to really physically separate those two cells from each other. So this is a picture of what the chromosomes look like. I'm going to draw a cartoon version on the board, but I want to just quickly do an overview of what we're going to be looking at. So this is G2 of interphase. In G2, every chromosome consists of two chromatids, genetically identical to each other. Remember, the goal is we're going to pull those apart. You can see that this centrosome is starting to grow microtubules. And as we enter prophase, that is really starting to set itself up. We're going to go through the major events of prophase, prometaphase, and then, metaphase, anaphase, and finally telophase and cytokinesis. So this is the cartoon version, and then that's an actual photo of this happening. So you can see the centrosomes. You can see that they've replicated in G2, so we now have two of them. And as we enter prophase, they're going to start migrating to opposite poles of the cell. I'm going to write all of this on the board, but I just wanted to show you this picture before I do my not-so-good picture on the board. But what you should really notice is that interphase, this is what the nucleus of the cell looks like. The DNA is all stretched out. You can't see individual chromosomes. Why do you think it's a good idea to have this DNA all stretched out and not tightly wound? Well, what is this cell doing mostly during interphase? It's doing its cell job, which requires enzymes, which requires the cell to make proteins. Proteins for export, proteins to use in the cell. In order to make proteins, that DNA recipe needs to be readily available so we can make messenger RNA and then translate a protein. So that's why that's why the DNA is all stretched out. As we start getting ready to divide the cell, though, it makes sense to have some nice, tightly wound, organized packages to pull apart. If you can imagine that this was all a basket of different yarns, and you wanted to pull out one piece of yarn and separate it from the others. It's going to be difficult if they're all bunched up like this. But if we really condense this and have it neatly packaged before we pull it apart, it's going to work a lot better. So that's what happens as we start leaving interphase and going into prophase. The way you know under the microscope this is happening is you can start to see individual chromosomes for the first time. And the reason for this is the DNA has condensed. What do I mean by that? What I mean is we have this long strand of DNA, And it's going to start winding around these little protein beads called histones. And as it winds and winds, it starts becoming condensed. So DNA condenses. And now, as we enter prophase, which is that first stage of metaphase, I'm sorry, what am I saying? The first, so DNA condenses, and as we start entering prophase, which is the first stage of mitosis, you start to see chromosomes, individual chromosomes for the first time under the microscope. Okay, so prophase, individual chromosomes are visible. As the DNA condenses. The other thing that's going to start happening is that nuclear envelope is going to start fragmenting and breaking down. So nuclear envelope breaks down. So let's draw that. Here's our cell. Here's our nuclear envelope. It's going to start fragmenting and breaking down. And meanwhile, we can see individual chromosomes for the first time. I'm just going to draw one pair of chromosomes. But these are tightly coiled. They're going to kind of pinch in the middle. You can't really draw that detail this small. And let's remind ourselves of a couple of things about these chromosomes in terms of structure. First of all, these are called sister chromatids, and they're held together by cohesin proteins. And on each sister chromatid, we have a little ring structure called a kinetochore. So if we blow this up, and we've done this before, remember this chromosome is actually going to constrict in the middle, and that region is called the centromere. But it's really just this tightly wound DNA inside here. It's not really inside something, I'm just kind of trying to replicate the shape here. Okay, and then cohesin proteins. And then this ring structure on each sister chromatid called a kinetochore. Remember, it's spelled chore, but it's pronounced core. This is what the microtubules are going to connect to on each sister chromatid. So microtubules are going to connect to the kinetochore on each sister chromatid. And in fact, those microtubules start emerging during prophase. So what happens is those centrosomes migrate to opposite poles. Okay, so centrosomes migrate to opposite poles, and microtubules start emerging. If you look at the cartoon version, you can see those microtubules are starting to emerge as these migrate to opposite poles. You know you're at the next phase called prometaphase when those have reached opposite poles and the nuclear envelope has completely disappeared. And now those microtubules are going to connect, they're going to attach to the kinetochore on each sister chromatid. And they're going to do this in a very specific way. They're going to do this so that each sister chromatid is connected to a microtubule from an opposite pole. So let's see what that would look like. So as we enter prometaphase, the second stage of mitosis between prophase and metaphase, The nuclear envelope has disappeared. So nuclear envelope gone. And those microtubules attach to sister chromatids. And remember, this is done in a very specific manner with each sister chromatid connected to a microtubule from opposite poles. So here, those microtubules started to emerge. And now they're actually going to attach to sister chromatids. So let's draw our pair of chromosomes again. Cohesin proteins holding them together. These ring structures called kinetochores, and that's where the microtubules are going to attach. They're going to attach in such a manner that each sister chromatid is attached to an opposite pole. This is purposeful. Okay, we're also going to have some microtubules that are going to not connect to kinetochores. So this is called a non-kinetochore microtubule. And the ones that are connected to a kinetochore are called kinetochore microtubules. Those are going to serve two different roles in dividing the cell. Now what happens is pulling. If you imagine that these two are connected to strings or ropes from opposite ends, and they're getting pulled, they're going to end up like this. And they're all going to end up in the middle of the cell. And as soon as that happens, we're at the next stage called metaphase. As soon as they're aligned in the middle, they're called metaphase. This imaginary equator of the cell is called the metaphase plate. So chromosomes align at the equator, in quotes, of cell. And it's called the metaphase plate. So I'm just going to draw that really fast. Here's our cell, no nuclear envelope. By the way, why do you think that nuclear envelope needs to break down? It needs to break down because those microtubules need to get in there to connect to the sister chromatids. So now we have the sister chromatids aligned in a very specific way. Each sister chromatid is facing an opposite pole because they've been pulled. Kinetochores. Each kinetochore must be connected to a microtubule from an opposite pole. And then we have the non-kinetochore microtubules. Those are going to start elongating the cell. So those non-kinetochore microtubules, those are going to grow and elongate the cell. The kinetochore microtubules are what's going to actually pull those sister chromatids apart. They're pulling. Now what happens is an important checkpoint. We want to make sure every kinetochore is correctly connected to a microtubule before we pull those apart. What if this was not? If this was not correctly connected, one cell is going to get too much DNA and one cell is not going to have enough. We don't want that to happen. So we're going to go through a very important checkpoint called the M checkpoint. It's also called the spindle checkpoint. If everything checks out and everything is correctly connected, we're going to have a cascade of events that lead to those sister chromatids separating. I want to talk for a second about that cascade of events because it's a very, very important example of an enzyme inhibitor. Remember, we don't want enzymes just being active all the time. Sometimes we want to control them, and we do that by inhibiting their action. there's a very important enzyme that's going to come in and digest those cohesin proteins. And the second those cohesin proteins are digested, we can pull these to opposite poles. They're not being held together anymore. They're already under tension, and as soon as there's not that sticky stuff holding them together, they're going to pull apart, and we're going to be at anaphase. This cascade of events is called the anaphase promoting complex. The reason I give you this detailed story in a non-majors class is because I think it's a very important example of enzyme inhibitors. So anaphase promoting complex. I want to see which of these works better. There we go. Okay, couple of important terms. Separase. I love this enzyme name. It might be one of my favorites. Okay, it ends in ASE, so we know that it's an enzyme. Separase is what's going to digest those cohesin proteins and cause those sister chromatids to separate. And separase is inhibited by an enzyme inhibitor called securin. I love this name too. Securin is what is keeping separase from being active. So this is an enzyme inhibitor. So separase is the enzyme that digests, hydrolyzes the cohesin proteins. Securin is the enzyme inhibitor of separase. So what happens is we have this M checkpoint. If everything's okay, then we're going to go through this cascade of events. So if M checkpoint, everything's okay, then we're going to go through this cascade of events. Securin is released. Separase is active. Separase now digests the cohesin proteins, and sister chromatids separate. The second those sister chromatids separate, we are in anaphase. In anaphase, the cell has been elongating due to those non-kinetochore microtubules elongating the cell. And now what happens is the kinetochore microtubules are going to start shortening and pulling those chromatids to opposite poles of the cell. So here we go. They were together, they've pulled apart. They're going to start moving to opposite poles of the cell as the kinetochore microtubules shorten and the non-kinetochore microtubules elongate. Okay, so chromatids move to opposite poles of the cell, and the cell elongates. And again, this is due to the microtubules. We know we're at the next phase, telophase, when those nuclear envelopes start to reform and essentially forming two new nuclei. Okay, so telophase, however you want to pronounce it. Two stages. One is that around these two new sets of chromosomes, one at one end of the cell and one at the other end of the cell, nuclear envelopes are going to reform. And that's essentially division of the nucleus. That's really what mitosis is, division of the nucleus. So new nuclear envelopes form. And now we need to go through cytokinesis. We need to divide that cytoplasm. To essentially divide that into two separate cells. How do we do that? It depends on if you're talking about a plant or an animal. Sorry, this is the cartoon version of interphase, then prophase, metaphase, anaphase, telophase. Cytokinesis. Animals are going to develop what's called a cleavage furrow, and plants are going to develop what's called a cell plate. So plants, cell plate. That cell plate is really just laying down new cell wall material. We're going to lay down new cell wall material in the middle here, and that essentially separates them into two separate cells. That's why plant cells have this almost brick-like uniformity. Animal cells, though, are going to pinch with something called a cleavage furrow. This contractile ring of actin is going to pinch that cytoplasm in half. This is what happens in animals. So this would be the cell plate in a plant cell. Start laying down the material, it eventually fuses together and eventually fuses with the plant cell wall to effectively divide that into two very uniform cells. And then this would be a cleavage furrow. A ring of actin, almost like putting an elastic band around your waist and pulling both ends, and it pinches that cytoplasm in half. This would be a microscope picture of the new cell wall material being laid down in a plant cell. These are actual microscope pictures of the stages of mitosis in an onion root. You will look at this in lab if you're taking the lab portion of this course. But I just want to point some things out. So in prophase, you start to see individual chromosomes, and they become very condensed by the time we get to prometaphase. Then at metaphase, they're aligned along the equator of the cell. It's really hard to see that here because what's happening in a plant cell, sometimes the chromatids are really long. So you get more of this sprawling rather than a nice little line. You get this sprawling effect. But this is indeed all aligned on the equator. These start pulling apart at anaphase. So they start looking a little more like this. And then at telophase, we see it starts to recondense and the nuclear envelopes have reformed. And you can see that cell plate material being laid down in the center here. So prophase, prometaphase, metaphase, anaphase. They've pulled apart and they're starting to move. This would be very early anaphase. They're going to keep migrating, and then we finally get to telophase, where they're, instead of being sprawling like this, they're more like this as those nuclear envelopes reform. The cell cycle, of course, is controlled by these checkpoints. These checkpoints are very, very important because without these checkpoints, we're not going to have normal cells. Remember that that G1 checkpoint is what keeps bad DNA from replicating. G2 checkpoint is going to make sure the cell is ready to divide, and M checkpoint is going to make sure every kinetochore is correctly connected to a spindle fiber, to a microtubule. When this checkpoint gets out of whack is when we get cancer.